So the next talk is by Eleonora Rachtman. She's going to be talking about their new method for using locality-sensitive hashing in contamination removal. So take it away.
Okay. Hello, can you guys see it? Okay, good. Okay, so hello everyone. My name is Eleonora and I'm a bioinformatics graduate student in Dr. Mira Rapp's lab at University of California, San Diego. So today I'm presenting on a topic of contamination removal using locality-sensitive hashing. So in recent years, there have been a number of reports pointing out to the presence of contaminants in public repositories. And sources of contamination are abundant. This pretty much could be anything upstream of sequencing, including sequencing itself. And normally contaminants should be dealt with either at the read level or at assembly level before they have a chance to propagate into public databases. So for purposes of our current study, we are aiming to deal with contaminants at the read level. So here is a problem definition. Our input is a set of sequencing reads that belong to target group, combined with a set of sequencing reads that belong to group of contaminants. And our output is a set of sequencing reads that belong only to target group. So how one can approach this problem? So normally, if you have a set of references, you can use them to construct your reference database, and then you can search your queries against your database to see whether a match can be found or not. If you know what your targets are, you can actually construct a reference database of your targets, and in this case, you will be retaining matched portion of your query reads. If you have an idea what your contaminants are, you can include them in your database, and in this case, you'll be discarding matched trees, and you'll be retaining unmatched portion of your queries. We do believe that this problem can be solved in alignment-free fashion. So all comparisons are made at the level of k-mers, so k-mers of references versus k-mers of queries. Obviously, this problem is very simple if you know, if you have references for your group of interest and you know exactly what you're looking for. However, it becomes much more complex if you don't have references available or you don't exactly know what you're looking for. So when we started working on this problem, we actually looked out there to see whether there is any publicly available software that could have been used to solve this problem. So first thing that comes to mind is, of course, Kraken. Kraken is a great species identification tool that allows you to use a set of references to construct lowest common ancestry database. And then you can query your sequencing reads again this reference database to see where the match can be found. Kraken calls a read classified if number of k-mers that match to the database is above user-specified threshold alpha. So we evaluated sensitivity of Kraken in our phylogeny leaf-out experiment. I just want to point out that this was part of our previously published work. So we started with a reference phylogeny of 10,000 bacterial and archaeal genomes. Since it's a phylogeny, it means that we know all provides distances between all the leaf, and we extracted query samples out of the set, and we used all remaining genomes in order to construct our reference database. We selected our queries such that we tightly control distance from a query to its closest species that we included in a database, and we also aimed to have a range of distances between query and the closest neighbor. In addition, we also added a set of sequencing reads from plants to our query set. The notion was that plants are actually substantially far from everything that we have in a database. So when you have a plant read being classified, it means that this is most likely a false positive. So setting this experiment up this way allowed us to evaluate recall and false positive rate in this experiment. So result is presented on the right side of the graph. So on the right side of the slide. So what we're looking at is x-axis is a distance from query to the closest species in a database, and on the y-axis we have recall. What we found that as long as query sequence is within 5% to the closest species in a reference database, we can still classify nearly 80% of the reads from the sample. However, performance deteriorates if query gets more far away from what we have in a database. None of the parameters, so we varied some of the parameters, but none of them really produced result which would be any different. So you can see that we always kind of have this characteristic elbow on a graph, unless you would be willing to go to substantially high level of false positives, which is probably unacceptable for majority of applications. And all of this experiment gave us an idea about our current project. So we were trying to see whether we're capable of expanding capabilities of existing software by relying on inexact k-mer matches between the read and a reference database. And again, just to underline, we believe that this is important in cases where exact reference assemblies for a group of interest are simply not available. So here is a high-level architecture. So we start with a bunch of genomes, we extract all the k-mers out of them, and we include them into our reference database. This is what happens at the map time. At the query time, we extract all the k-mers out of our query, and then we search them against a database. And then if hit as found, we actually compute Hamming distance between the database k-mer and the query k-mer, just to confirm. So if Hamming distance is within the user-specified threshold, we call this k-mer a match. However, as you might imagine, we have tens of thousands of reference genomes that we might be dealing with, which produce billions of k-mers. So clearly, we need some meaningful way how we can organize these k-mers in our database so we can search them faster. And this is where locality-sensitive hashing portion comes in. So locality-sensitive hashing, unlike standard hashing, when you're trying to differentiate between genomes, allows you to hash similar items into the same bin with high probability. So what does this really mean for us in terms of k-mers? So this actually means that if you have a k-mer and you can subsample certain positions in this k-mer to generate its short representation. And if you have a pool of k-mers for which short representations are identical, you can record information about such k-mers in the same row of your indexing table. And in practice, this procedure, of course, needs to be repeated multiple times. So in practice, we are dealing with multiple index tables. And here is a more detailed view how everything comes together in practice. So we start with a k-mer, we subsample certain positions in the k-mer to generate our short representations. We actually call them signatures. And again, we can have multiple of them, and then information about each one will be recorded in a corresponding index table. We also have a pointer from index table to our k-mer encoding array. So this one contains information about binary representation of original k-mer. And this is what allows us to compute Hamming distance exactly between a database k-mer and a query k-mer. So we can actually confirm that the match that we found is not a false positive. There are many parameters that you can vary when you design your LSH. In our system, these are primarily k-mer lengths, number of positions that we can select, subsample out of a k-mer, or of course, number of signatures that we can produce, we can generate. So I want to show you a couple of examples in our simulation. So in this case, number of subsampled positions we denote with letter H, and number of hash functions, which is tantamount to number of signatures, we denote with variable L. So here is an idea. If you have two k-mers, which are reasonably close to each other, it means that their Hamming distance is actually very small. You would hope to get your expected number of k-mer matches to be high. So that effectively, if you're on the left side of the graph, you're trying to maximize your function in this area. At the same time, if you have two k-mers which are distant from each other, you would want to minimize number of expected k-mer matches in this region. So if you're on the right side of the graph, this is what you're trying to do. Now, as number of signatures that we extract out of a k-mer increases, so here we're showing what happens if we go from L equals 1 to L equals 2, expected number of k-mer matches is also going up, and this makes sense. However, it cannot be increased infinitely because this also generates substantially larger footprint for our database. So after titrating these parameters in practice, we actually settled at L value equals 2 as a default parameter for our software. In a similar example, we also modeled what happens if you pull different number of positions out of original k-mer. So in here, we're showing what happens if you go from 15 to 6. So as your shorter representation is getting shorter and shorter, expected number of k-mer matches is also going up, and this is what we observed. However, and this is nice because we're in a region where we want to maximize it. However, problem is that in region where we want to minimize it, it also goes up. So again, another example that when we kind of titrated it in real life, we settled at H value equals to 15 for kind of default setting of our software. So clearly it all indicates that parameters of LSH needs to be carefully chosen and titrated in practice, and this is what we did for our software. So our algorithm is implemented in a software called Consult, which is publicly available on GitHub, and we evaluated performance of our software in 4 experiments. So we started with sensitivity analysis. The design of this experiment was very similar to the ones that I explained before. So what we basically found is that as long as query sequence is within 15% to the closest species in a database, we can still match nearly 80% of the reads from such sample. Again, performance of Kraken was slightly worse in this case. Here, I also have to point out that level of percentage of false positives was actually quite similar, comparable between 2 pieces of software. We also tested other tools that were mentioned in the literature that could have been used for this purpose. However, nothing really produced results any better than Kraken or Consult in this particular setting, unless, again, you would be willing to go to unacceptably high level of false positive, which is probably not an option for majority of applications out there. So in our second experiment, we evaluated effect of database composition itself. So we constructed reference database using references from Tree of Life. And as queries in this experiment, we used marine microorganisms that we actually have got from reference number 4, publicly available set. Marine microorganisms are considered rare and rare species, and they are known to be poorly represented in public repositories. So in a way, we treated them as kind of novel, most challenging queries that you might potentially expect to see in real life. So on x-axis of the diagram, we're looking at percent reads match, and on the y-axis, we're looking at percent query genomes. So basically what we learned is, for example, for Consult, for 50% of the queries, we can match at least 50% of the reads, and performance for Kraken was kind of slightly worse. When we explored 2 other data sets, we actually found that relative performance was about the same. However, absolute performance of LSH in this particular case was actually heavily guided by database composition, and this makes sense. We found that the best performing database was GTDB, and this makes sense because this is the most diverse data set out of all 3 that we tested, and you can see kind of level of performance on the graph. In our third experiment, we evaluated ability of Consult to remove contaminants from real data. So for this experiment, we used the data set from Miller's paper. In this study, authors used a combination of short and long reads to generate 15 novel Drosophila assemblies. So we use their data set to compute all pairwise distances between Drosophila species, and then we calculated relative error in distance estimate before and after filtering. So on the graph on x-axis, we're looking at all pairs of Drosophilas that we evaluated, and the y-axis, we're looking at change in relative error. So here is what we learned. We learned that before filtering, distances are very frequently underestimated, which makes our relative error negative. At the same time, after filtering, distances are usually positive. They might get overestimated. And for a couple of samples that we denoted with a different color, we actually overfiltered, so distances have got slightly worse. However, for majority of samples, you can see that we're doing kind of quite a good job. Overall idea is, of course, you're trying to minimize your relative error, so you're trying to get as close to 0 as possible. And you can see that we kind of, especially for some of the worst offenders, we're doing a very decent job cleaning them up. So this was a positive result. And in our final experiment, we evaluated ability of Consult to separate nuclear reads from organelle reads for purposes of organelle assembly. So we used a data set from Denmark sequencing project. So in this study, authors attempted to assemble 210 mitochondrial genomes. So 18 species failed in assembly. 8 species produced poor quality assemblies. And everything else was good. So out of their good assemblies, we basically randomly selected a set of 18 to use as a positive control in our experiment. On x-axis, you're looking at mitochondrial genes that you would expect to see annotated if your mitochondrial assembly is complete. I have to point out that in original studies, they used seed and extent method, meaning that assembler was actually relying on finding a seed, which is Fox gene in this particular setting, and then was trying to expand from there. Yeah, and in first part of our experiment, basically what we did, we took original assemblies produced by the study and just pre-annotated them. In the second part of this experiment, we used it was, we compared seed and extent to standard coverage-based method. So coverage-based assemblers are aligned on difference in coverage between nuclear reads and organelle reads. So normally you would expect your organelle reads, of course, having much higher coverage. And you can see performance was actually, well, borderline. And in our final step of this experiment, we actually attempted to use our chimer-based approach. So what we did, we constructed a reference database using all mitochondrial genomes available in RECOMB-Seq, and then we queried raw data against our database to extract just mitochondrial portion. And then we applied standard whole genome assembler to assemble just this mitochondrial portion. So we essentially managed to rescue about 9 out of 18 samples that failed in the original study. We produced decent quality assemblies for poor kind of quality ones. And we did as good of a job as the original study on a good quality assemblies. Here I also have to point out that our chimer-based approach was actually about 7 times faster than coverage-based, primarily because here filtering does a lot of heavy lifting, which makes our assembly step very fast because it deals only with a small portion of sequencing reads. And with this, I kind of want to wrap up. I just want to point out that we believe we developed a general purpose algorithm that allows to test read membership, and we're hoping that it may be useful for majority of applications. So thank you very much.
Thank you much for the great talk. We do have a couple minutes for questions, and there's a couple in the Q&A. I'm hoping we can get to both of them. The first one is from an anonymous attendee. I'm wondering if the genetic material in the species that were recently horizontally transferred from a distant species could be discarded by this method, especially when a large gene cluster was transferred by plasmid conjugation.
I guess it can be, however, so it would probably require some pre-processing of genomes to make sure, because we're operating at the chimer level. So I think some of the chimers in this case needs to be discarded, right? And then we probably will be relying more on kind of discriminative chimers. What I'm not sure about is how it will affect regions of similarity, because if you're trying to estimate distances between 2 species, right, you're not only trying to label them, you're trying to get all the reads that are actually common and not common between them. So because commonality gives us appropriate distance. So I guess we can, but short answer is yes, we can, but we would need to do some pre-processing of the data.
Very good, thanks. And then the second question is from Bara Arabi. Great talk. Do you think the exact design of the spaced chimers, how close the gaps are in sample positions, affect the performance? And if so, how? That would be added part's mine.
Yes, so it's actually a very nice question, something that I was thinking about myself this morning. So if we're in the current version of the software, what we're doing, we're actually extracting all the chimers, so all overlapping chimers, which makes our chimers that extremely large. Luckily, we found that in order to basically classify our reads, we actually don't need all the chimers, so we can get away with only a subset of chimers, which we are including in our databases because we have a limited capacity. But yes, if we find a way to, for example, spaced chimers, right, to separate basically meaningful chimers from chimers that don't really give us any additional information about species, this would allow us to do a lot of good things and certainly, much more meaningful work. But for now, this is kind of not something that we're doing, but it's a good question.
Good. Thanks again for the great talk. That's perfectly timed as well.
Thank you.
So with that, we're going to move on to the final talk of the session.